our mechanism is the GPCR, Phospholipase C protein kinase C cascade, which is a mechanism that triggers the release of calcium ions through the release of a secondary messenger cascade. The physiological importance of G protein receptors is relevant due to the binding of calcium to myosin, which triggers muscle movement because of the usage of ATP.
To begin the signaling transduction pathway, the ligand binds to a 7-transmembrane domain called the GPC receptor. As a result of the binding of the ligand, this will induce a conformational change of the receptor and increase the affinity for the heterotrimeric G protein, which composed of an alpha, beta, and gamma subunit. The G protein will then bind to said receptor due to the increased affinity, and the alpha subunit will then experience a conformational shift. The G alpha subunit will then release its GDP, which is subsequently replaced by GTP.
This causes a loss of affinity for the beta and gamma subunits and promotes association of the alpha subunit with the PLC. The activated PLC will then cleave the PIP2 complex into secondary messengers IP3 and DAG. Simultaneously, the GTP on the alpha subunit is hydrolyzed into GDP, and the alpha subunit rebinds with the other subunits to reform the heterotrimeric G protein complex. The PLC complex is then inactivated. DAG remains bound to the phospholipid bilayer via London dispersion forces and also experiences an increased affinity for the PKC complex, which in turn allows them to bind.
IP3 then binds to the IP3 receptor, located on the extracellular membrane of the smooth ER, and allows for the release of calcium ions into the cytosol. Calcium ions act as a secondary messenger, binding to the PKC-DAG complex and increase its activity. Calcium ions will travel to other parts of the cell to be utilized in other physiological processes.
This project was made by Valerie Hawking,
Lacey Jackson,
Sarah Cyani,
Kristen Luongo,
and Elisa Medina. If you enjoyed this video, please be sure to give us a thumbs up on YouTube and subscribe!